Hello, welcome to Chapter 3. Chapter 3 has prokaryotes and eukaryotes in it, and so we'll have two videos on prokaryotes because this is a microbiology class, and I won't do the eukaryotic video, but you can just read through that because there's not a lot of eukaryotic questions on the test. Okay, so what does it mean to be alive? Well, there's 5 processes that define life. So if you're any living thing on this planet, anyway, you share 5 things in common, these 5 processes. So growth. Living things can increase in size. So you started out as a single-celled zygote, and then you grew into embryo, an infant, and an adult. Now, bacteria can increase in size, but in a microbiology class, if you're talking about bacterial growth, you will always be talking about an increase in bacterial number. So that's really important to note. While bacteria can grow a little bit in size, if you're talking about bacterial growth, that's growth in their number. If you are living, you reproduce. You increase the number of you, and you can do that sexually or asexually. You are responsive to your environment, so as your environment changes, you respond to that. If you're living, you do metabolism. So if you're a living thing, you take in nutrients and you use those nutrients in chemical reactions to grow, reproduce, and respond, the 3 previous processes of life. You have a cell structure. All living things are made of cells. Whether you're a single cell or you're a multicellular creature, you are made of cells. So you could call the cell membrane, you can call it the plasma membrane, the cell membrane. What it actually is a phospholipid bilayer. You probably remember that from your previous classes. But it gives the cell a structure. That membrane gives the cell, and other things help give the cell a structure. In addition to your phospholipid bilayer called a plasma membrane or a cell membrane, some cells also have cell walls that also help maintain its shape. And this is just if you bought the book, this is table 3.1 from your book that summarizes characteristics of life and their distribution in the microbial world, including viruses. So now let's do a little evolution. There's 3 evolutionary domains. So these are the domains. The domain bacteria, which is what we will be mostly interested in this class. The domain archaea, which are microscopic creatures, and cells and single cells, but they don't really cause us disease. They don't cause human diseases, and so we won't study them that much. And then the eukaryotes, or the domain eukaryota, that's where we live. So cells, if you're talking about cells that we're interested in as humans, those would be the bacteria cells and the archaea cells. Those are the prokaryotes. And then the eukaryotes, those are our cells. So here's a cartoon of this top one. Is a cartoon of a prokaryotic cell, and TEM stands for transmission electron micrograph, and this is actually a colorized transmission electron micrograph of a prokaryotic cell. And this, the bottom cartoon is a cartoon of well, it's actually a transmission electron micrograph that's been colorized like a cartoon to show a eukaryotic cell. And you can see that eukaryotic cell is, just by glance, it's more complicated than the prokaryotic cell. So if you heard of this thing, the cell theory, you've probably heard of like the theory of gravity, the theory of evolution. Well, there's a cell theory. Cell theory states that all living things are composed of cells. And we talked about this last slide. All cells have a phospholipid. This just says lipid, but for our cells anyway, it's a phospholipid bilayer. aka the plasma membrane, or PM. It allows the cells to grow, reproduce, respond, and metabolize. This cartoon on the left is an ostrich egg. It's 13 centimeters in diameter. So, and then it shows this little spot right here. So it shows the size of an ostrich egg, and then it shows you in relation, here's the size of a virus in relation to that ostrich egg. Here's a bacterium, Staphylococcus, a micrometer in diameter. And then, I don't know if anybody hikes, but sometimes if you drink the water out on a hike, you can get this parasitic protozoan called Giardia. And now in this cartoon on the right side, the transmission electron micrograph that's been cartoonized and colorized, it gives you some labels. So for example, this piece is the nucleoid. That's where, so it's not a nucleus because it's not surrounded by a phospholipid bilayer, but it is a nucleoid. That's where bacterial DNA are. Ribosomes, where you make proteins. The cytoplasm, so all this is the cytoplasm. And the glycocalyx, calyx in Greek means sugar cup. So it's the sticky outer covering on the bacterial cell. And then deep to the glycocalyx is the cell wall. We'll look at that in detail. And then deep to the cell wall is the cell membrane. And then here's a flagellum to help the bacteria move in space and time. And then inclusions, those can contain different things in a bacteria. So prokaryotes are 1 to 2 micrometers in size versus eukaryotes can be 10 to 100 micrometers in size. So a eukaryote can be 100 times the size of a prokaryote. Prokaryotes differ fundamentally from eukaryotes in their ribosomal RNA sequences, the kinds of lipids in their plasma membrane, and then also their cell walls, the chemistry of their cell walls. Prokaryotes are found in the domain bacteria and the domain archaea. They have no nucleus, but this is tricky. Don't be tricked on this. They do have a nucleoid where the DNA is located. Prokaryotes have no membrane-bound organelles, so they don't have, for example, a Golgi apparatus or an endoplasmic reticulum or a nucleus. All those are membrane-bound organelles. Up here in the corner, this is a scanning electron micrograph that's been colorized of bacteria. So that's what they look like in a scanning electron microscope. Importantly, scientific nomenclature. How do you write out the names of bacteria, the genus and species? So you probably know our genus and species is Homo sapiens, and so bacteria have a genus and species too. The first letter in genus is capitalized. So for example, Clostridium botulinum, that causes botulism. The C is capitalized, and both words, Clostridium and botulinum, are italicized. So you should learn how to write out scientific nomenclature for bacteria. So the correct order of a complete scientific name of bacteria, Escherichia coli, they just abbreviated that, E. coli. This is E. Escherichia is the genus, coli is species. And then there's this part, O157 colon H7. That's the strain of bacteria. So they get a little extra in their scientific nomenclature compared to us. So bacteria have what's called an envelope, and actually so do viruses have envelopes as well. But bacterial cell envelope is composed of these three structures. So the cytoplasmic membrane, or the cell membrane, here goes all the way around. Then superficial to the cytoplasmic membrane is the cell wall. So cell wall would, I don't know if I can draw very well. This would all be cell wall. And then superficial to the cell wall would be the glycocalyx. Inside the bacteria, you should know there is a nucleoid. The whole thing is full of cytoplasm. The ribosomes, that's where the proteins are made. And then there are these inclusions. This is a cartoon of a phospholipid bilayer. So it says phospholipid bilayer over here. Here's the hydrophilic head and the hydrophobic tails on the external surface. Here's a hydrophilic head and the hydrophobic tails on the internal surface. That's the bilayer, the bi part of the bilayer. And then each one of these little guys with the heads and the tails, that's a phospholipid. And then you can stick these integral membrane proteins within the phospholipid bilayer. If they go through the phospholipid bilayer, they're an integral protein. If they're just on the inside or just on the outside, they're a peripheral protein. Here's another integral protein that has a little bit different shape than the first one we looked at. So this is the cytoplasm is inside the cell. Okay, and we talked about this. It's the cell membrane or the plasma membrane, and what it's composed of is a phospholipid bilayer. Archaea, in the domain Archaea, prokaryotes in the domain Archaea, they lack these phosphate groups, so they're a little different in that respect. Some bacterial plasma membranes or phospholipid bilayers contain these sterile-like molecules called hopanoids, and when you grind up the bacteria and look at it biochemically, you can find those. So what does the plasma membrane do? What does this phospholipid bilayer do? It separates the ICF. I call the ICF, that's everything, the intracellular fluid, that's on the inside of the cell, and then the ECF is the extracellular fluid on the outside of the cell. The plasma membrane, phospholipid bilayer, it controls passage of materials in and out of the cell. So if you're a big old molecule that's hydrophilic here, you cannot just go through the phospholipid bilayer, no problem. You might be able to get through a channel in one of these integral membrane proteins, but it's controlled. So the cell controls what goes in and out of it. So, and there's a term for that. It's called that the plasma membrane is selectively permeable. So permeable just means you can get through something, and selective means only certain things can get through the phospholipid bilayer. Large molecules can't get through the plasma membrane. Ions, sorry, this is written over, but that word is ions, are repelled by the phospholipid bilayer. If you are a hydrophilic or water-loving substance, you cannot cross very easily unless you go through a channel. So the cytoplasm of prokaryotes, you have the cytosol, that's the liquid portion. It's got all these things in it, water, ions, etc. It's got the DNA and the nucleoid region. We talked about that. Inclusions in a bacteria might be just a reserve of lipids, that's fat. You could have starch, you could have nitrogen in the inclusion. You can have PHB, which is a molecule that you can use to make plastic. You could have these sulfur inclusions. So each one of these guys is a rod-shaped bacteria, and these black dots inside, those are inclusions, and they contain sulfur. You can also, if you are cyanobacteria, that's a blue-green photosynthetic bacteria, your inclusions could be full of gas. So here's bacteria, if it's got a lot of gas in its inclusions, you call it a gas vesicle, and it helps bacteria float. So that's kind of cool. It's a flotation device. There are magnetotactic bacteria, and they've got magnetite in them, so the bacteria can orient themselves along the pole, and that's what this is. So here's some magnetite. It's going to align this bacteria to the North and South Pole. Ribosomes, we talked about that. It's really important to note that in bacteria, the ribosomes are 70S, and in eukaryotes, that's our cells, the ribosomes are 80S. The ribosomes are made of polypeptides, so poly means many, peptide means protein or a short protein, and ribosomal RNA are RNA. Scientists can target the 70S ribosome with antibiotics to kill the bacteria. And then if you're targeting a 70S ribosome, that chemical won't target the 80S ribosome and will kill the bacterial cell, but not you. So an example of that is a drug called erythromycin. Probably you've heard of that. Prokaryotes, just like eukaryotes, they have a cytoskeleton that help the bacteria maintain its shape, made of actin. Most prokaryotes, so that's bacteria, which is what we're mostly interested in this class, have this thing called a cell wall. So in this cartoon over here, here's the rod-shaped bacteria. So you can see they cut this rectangle out of the rod-shaped bacteria and they enlarged it in the cartoon. And all of this piece, this kind of brick color, that's all cell wall. And it's got a really complicated structure that we'll look at in a minute. But the cell wall protects bacteria from osmotic forces. So remember, osmosis is the movement of water down its concentration gradient and through a permeable membrane or semi-permeable membrane. The cell wall helps bacteria attach to other cells, and that cell wall helps the microbes in their battle to not be killed by our antibiotics. Cell wall also gives structure and shape to the cell, bacterial cell, different shapes. So if you have a spherical shape, you're called a cocci. The single term is coccus. If you are a chain of cocci, in your bacteria, you're called streptococci. If you are a cluster, not a chain, but a cluster of cocci, then you are staphylococci. If you're rod-shaped, like in the cartoon up here, the rod-shaped bacteria, that's called a bacilli. Singular is bacillus. So a chain of rod-shaped bacteria is called a streptobacilli. And then you can also have spiral-shaped bacteria. So here's some cartoons. So this would be a coccus, a single coccus. This would be a chain, streptococci. Here's a cluster of cocci, so that'd be staphylococci. Okay, so let's look at the structures in a little more depth. So the glycocalyx, remember that sugar cup, so it's sugary, that means it's sticky, so it allows bacteria to stick or attach to the surface of interest, which could be your mucous membrane. And glycocalyces also allow groups of bacteria to stick to each other. That's called a biofilm. So if you ever wake up in the morning and you feel your teeth with your tongue and it feels slimy, that's a biofilm. It can contribute to bacterial diseases. You can also get a biofilm on plastic medical equipment, so it's really important to use sterilized plastic medical equipment. And when you see pond scum, oftentimes that is a biofilm of bacteria. The glycocalyx helps the bacteria survive. It helps the bacteria avoid phagocytosis by the cells in our immune system, like macrophages. It also helps the bacteria to not dry out. Eukaryotes also have glycocalyces. Our cells also have glycocalyces. And glycocalyces, you can get them in two flavors. You can be a glycocalyx that is a capsule. That means it's firmly attached to the cell wall. Or you can be a glycocalyx that's a slime layer, and that glycocalyx is loosely attached. So here again, transmission electron micrograph of a capsule, a glycocalyx that is a capsule, so that's a firm attachment. And this is transmission electron micrograph of a staphylococcus. So you can see it's a circle, so it's a coccus. And then this glycocalyx, looks like this is a transmission electron micrograph that's not colorized. And this is a slime layer, so it's loosely attached. You can see that here's all slime layer. It's more loosely attached to the cell wall. And this is a transmission electron micrograph of Prokaryotes. That's fecal bacterium. Like I said earlier, you have cells in your immune system called macrophages, and they don't recognize a glycocalyx. So if the macrophage is moving around in your immune system trying to find bacteria to eat, so phage means to eat, destroy, and that bacteria has a glycocalyx on it, the macrophages won't recognize that bacteria and they won't destroy it. There are these receptors, receptors on macrophages will usually recognize this thing called a PAMP, pathogen-associated molecular pathogens. And the glycocalyx will be on top of the pathogen-associated molecular patterns, and it will hide, the glycocalyx will hide the PAMPs, so the macrophages with their tall-like receptors can't recognize it. When the microbe is, for example, the bacteria is dividing, for a moment in time, it doesn't have a glycocalyx, and then that's when the macrophage can recognize the PAMPs, and then they can eat the bacteria. Okay, more external structures of prokaryotic cells. So in this cartoon, this is a bacillus, and it's called a gram-positive bacteria. And we'll do gram staining. It's a basic stain that you do in microbiology, but gram-positive bacteria have a flagella that looks like this. So here's let's start deep. Here's the cytoplasmic membrane, the phospholipid bilayer. You can see the hydrophilic heads and then the hydrophobic tails. And you can see the bilayer nature of the cytoplasmic membrane. And then you have this cell wall made of this protein called peptidoglycan. We'll look at that in more detail later. But then here's the flagella that we're interested in. So it's kind of cool mechanistically if you're into mechanical things and like to think how they work. You don't have to know this detail for the test, but you got these protein rings, and then you have a rod of the flagellum. It's all embedded in the plasma membrane and the cell wall. And then here you have the filament of the flagella, and you can see the direction. These arrows show you the direction that the flagella rotates. Why do bacteria or prokaryotic cells in general want a flagella? Well, sperm have flagella too, and that's a eukaryotic cell, but you use the flagella for motility. So the most common structure that bacteria use is a flagella. Not all prokaryotes have flagella, however. Okay, on the previous cartoon, we looked at a gram-positive bacteria's flagella. This is a gram-negative's flagella. And you can see it's a little more complicated. It's kind of cool. You don't have to know the details, but again, here's the cytoplasmic membrane, the phospholipid bilayer. Here's the cell wall, and then superficial to the cell wall, or part of the cell wall, but superficial to the peptidoglycan layer, is this thing called an outer membrane. And you can see the outer membrane is not a it's not a phospholipid bilayer like the inner cell membrane is. But then you have all these pieces embedded in the membrane, the plasma membrane, the cytoplasmic membrane, and the cell wall. And then you have this hook portion and the filament portion of the flagella. And you can see that arrow shows the direction of movement. And then also, really importantly, actually, in the outer membrane of the cell wall of a gram-negative bacteria, you have this thing called LPS, lipopolysaccharide, and this thing is really deadly to humans. So we'll talk more about that, but it's kind of interesting. Flagella. What kind of flagella can you have if you are a bacteria? Well, you can be monotrichous. They have a single polar flagellum, so it's only at one pole. So this pink thing, this bacteria, at one pole, north or south pole, you have a single flagella, that's monotrichous. An example would be Vibrio cholera, the bacteria that causes cholera. And then you have this, the next type is Lophotrichus, or tufts of flagella. So that's this cartoon, tufts of flagella, and you can see them here. Pseudomonas fluorescens is an example of a bacteria that has tufts for flagella. And then Amphitrichus, so amphi, that's like amphibian. Remember, amphibian means it can be both in the water and on land. So these flagella are pretty much all over the bacteria, Amphitrichus. And then Paratrichus, you have, well, oh, sorry, I did that backwards. Amphitrichus is over here, is D. They have a single polar flagella. And then peritrichus. So peri just means surrounding. So the flagella are surrounding the bacterial cell. Here's some, these are scanning electron micrographs. So here's peritrichus. You can see there's flagella everywhere you look on this bacteria. You can see peritrichus flagella. Here's some tufts. So they're just kind of here. And then here's a polar flagella. So it's just one flagella at the pole. This is an interesting bacteria. I like this guy, Treponema pallidum. That's the bacteria that causes syphilis. So it's got this thing called an endoflagella. So the flagella is actually inside, in here, and it's made of an axial filament. So the internal flagella, like I said, it's called an endoflagella. And it takes the form of an axial filament. It wraps around the cell. If you are a spirochete bacteria, you have this endoflagella, and it causes, when the flagella moves, you move in the, if you're a bacteria, you move in this corkscrew motion. Also, the bacteria that causes Lyme disease, Borrelia burgdorferi, that also has an endoflagella made of axial filaments. This is kind of cool. So here's bacteria. If it wants to move, it kind of lines up. So this would be Amphitrichus. It lines up all flagella, and they're all moving in one direction. And zip, it'll go here. And then if their flagella don't all line up in one direction, it just kind of tumbles around, and then it will run here when it layers all its flagella back. And what it's doing is trying to get over here to an attractant. So the attractant could be light, it could be a sugar molecule, something like that. Okay, now don't get these two confused. There's flagella, and then there's fimbria, or fimbriae. Fimbriae is plural. So those are sticky. Again, bacteria like to stick things. They're bristle. They're like bristles, like hair bristles, and they're projecting from the bacterial body, and they help bacteria stick to other surfaces and also to each other. And you can see that fimbriae, these are these little short things, and then the flagella are longer than the fimbriae. So fimbriae, glycocalyces, they're important sticking tools. So whether bacteria is sticking to your cell or they're sticking to each other in a biofilm. And this is this must be doesn't say if it's a scanning electron micrograph. I think it probably is, but it's a micrograph of Neisseria gonorrhea. More external structures of prokaryotic cells, a pili, or pilus is singular. It's a special type of fimbriae, so a pilus is a kind of fimbriae, and it's used for this process of conjugation. We'll talk more about that later, but conjugation is what bacteria use to transfer DNA segments from one cell to another. Now, it may seem real sexy to look at this. There's a conjugation pilus between these two bacteria, specifically Salmonella. But it's not sexual reproduction. Sexual reproduction is what humans do. So you have a haploid sperm and a haploid egg. That's not happening here. These two bacteria are just swapping DNA. So it's not sexual reproduction. Some bacteria don't have cell walls, like Mycoplasma pneumoniae. There's no cell wall at all. There's no outer membrane. There's no peptidoglycan layer. It's real tiny. So this is each one of these Mycoplasma pneumoniae. They're real small. Originally, I thought they were a virus. Okay, if you don't have a cell wall, it shows in this cartoon, like mycoplasma and animal cells, like our cells, we don't have a cell wall. Water can move osmotically down its concentration gradient. And then in the bottom cartoon, it says cells with a wall, like plants, fungal cells, and bacterial cells. In an isotonic solution, it shows that water can still move in and out. But it will move out in an equilibrated fashion because this is an isotonic solution. So isotonic just means that the concentration of these molecules, the water molecules out here, or a solute molecule, is the same. Iso is the same as the concentration inside, and it's the same with this cartoon. So both these cartoons are showing a cell, like a eukaryotic cell, in an isotonic solution, and a bacterial cell in an isotonic solution. If you are in a hypertonic solution, so hyper means too much, that means that you have more solute out here in the solution versus the solute concentration inside the cell. And it's the same thing down here. You have more solute, these little dots. You have more solute concentration outside, hypertonic, outside the cell than you do inside of the cell. That means water wants to move down its concentration gradient. So for example, if you have a whole bunch of solute particles out here and fewer solute particles inside the cell, that means that you have less water, because you have more solute, you have less water on the outside, and you have more water on the inside, and water wants to move down its concentration gradient, so it moves out of the cell and into the solution in both the eukaryotic cell and the cell with the cell wall. What that does is it causes, when water rushes out of the cells, it causes the cells to shrivel. So we have isotonic, hypertonic, and now we have hypotonic. So hypo means less than or below. So in both of these cartoons, there's less solute molecules outside the cell, more solute molecules inside the cell. What that means is if you have less solute molecules outside the cell, that means you have more water outside the cell relative to the inside of the cell that has more solute particles and less water. So water moves down its concentration gradient in both conditions.